Okay, so chapter 20, it's about antimicrobial drugs. Here at the beginning of this presentation, we have one of the most feared microorganisms in the hospitals is Pseudomonas aeruginosa, and it's highly feared just because it has a lot of genes that makes it resistant not only to antimicrobials but also to disinfectants. So we're going to see part of the history of the development of antibiotics, including a couple of key researchers, Paul Ehrlich and Alexander Fleming. And then we're going to name some of the microorganisms that produces antibiotics. So back in early history of humans, we had a lot of death due to infectious diseases. So unfortunately, many of them could be not only preventable but also treatable. And then with advancement of science, there was some minds that were thinking, well, I mean, what if we can have a chemical that can kill microbes but do not affect the host? Because they started to experiment with some antimicrobial substances and they found a lot of toxicity. So then selective toxicity is the property of only affecting one organism while protecting the other. And then, of course, in the case of antimicrobials, what we're looking is to destroy only pathogens by not damaging the host cells. And then any treatment for any sign and symptom or disease is known as chemotherapy. That doesn't necessarily need to be cancer. And then antibiotics are substances that are produced by microbes naturally that they use to antagonize other microbes in order to survive. And then we have specific antibiotics that are produced artificially or synthetic that are known as antimicrobials. They interfere with the growth of microbes. You saw at the beginning of the semester how Alexander Fleming was studying his plates with Staph aureus, and then he saw absence of growth in one of these plates, and then he always saw a large colony that had a clear halo and no growth of microbes around this colony, or small colonies that were growing around this mold, but they weren't the normal size. So then he discovered the mold fungus that produces penicillin, which was Penicillium notatum. And that was in 1928. And then they started to produce penicillin. And then they also used for certain microbes like Streptococcus a dye, which was called prontosil dye, red dye, which is a sulfanilamide. So this was used to treat streptococcal infections. Now, the discovery of penicillin and the production started early. However, it was released for clinical trials in the 1940s. And it was found out that it was pretty effective antibiotic. So here we have an example of naturally produced antibiotics released into the surface of the agar plate and how you have microbial antagonism. In this case, we have these large colonies with irregular shape, irregular borders. Some of them are unbonated, as you can see this dimple in the middle. So this is a contaminant fungus. And then all of these small colonies, beige color, these will be the actual normal bacteria plus other contaminants here. And all of this clear halo basically is telling you that there is secretions of antibiotics that prevents the growth of bacteria around. So that's normal antibiosis. And then most of these antibiotics has been found to be in nature, produced by different microorganisms. So here we have the table showing you these different types of microorganisms and what they produce. So we have some gram-positive rods like Bacillus subtilis, or now it's called differently Spiricisteni. It produces bacitracin. We have Panobacillus polymixa that produces polymyxin. So these two, together with the one that Streptomyces fridii produces, which is neomycin, it is combined to make an unguent that it is sold on, in the countertop. So this is a topical combination of antibiotics. So it is used in the skin. And then we have some members of the Actinomycetes family, which are Streptomyces. We have Nodossus, Venezuela, Arophacians, that produces amphotericin B, chloramphenicol, chlortetracycline, and tetracycline for this last. And then Saccharopolyspora erythraea produces erythromycin. And then Streptomyces griseus produces streptomycin, and Micromonospora purpurea produces gentamicin. So those were bacteria. And now for fungi, we have Cephalosporium species. Remember, this abbreviation SPP means species. We have Penicillium griseofluvum, and then we have Penicillium chrysogenum. So this one, Griseofluvum, produces Grisofulvin, and then Chrysogenum produces penicillin, and then Cephalosporin species produces different cephalosporin, but one of the examples will be cephalotin. That's okay. So now let's move into some problems of using antibiotics. And then we're going to define what is a spectrum, so classify antibiotics based on that spectrum, and then define what is superinfection. Okay, so a spectrum refers to the ability to kill microorganisms in a particular number, or the number of microorganisms that antibiotics can destroy. So we have narrow spectrum antibiotics and we have broad spectrum antibiotics. So narrow spectrum antibiotics basically will be the ones that only affect very few species. And then broad spectrum covers a lot of species, including gram-positive and or gram-negative bacteria. And then superinfection, it is an abnormal growth of bacteria that it is resistant to antibiotics. So example, we have Candida albicans and Clostridioides difficile, or C. diff, or colloquial term. So unfortunately, one of the disadvantages of using specific antibiotics, especially those ones that are broad spectrum, it is that you not only kill the pathogen, but also destroy the normal flora. And then some of the members of the normal flora, like C. difficile, can overgrow much faster than the rest of the normal flora, can colonize really easily the epithelium of the large intestine, and then when it does, it creates like a membrane that prevents the absorption of water, and then the person will have diarrhea. In the case of Candida albicans, I mean, this is not part of the normal flora. It's actually a pathogen. It's a yeast, and then of course these yeasts do not colonize unless the person has an ulcer, inflammation, or it lacks this normal flora, and that will affect the throat, the mouth, the vagina, and even sometimes up to the esophagus. So here we have a table showing you some antibiotics in their spectrum and what do they cover. So we have antibiotics that can kill prokaryotes, that are pathogens, of course, and then we have some that can kill pathogens that are eukaryotes. Classification of prokaryotes: mycobacteria, gram-negative bacteria, gram-positive, chlamydias, and rickettsias, which, if you recall, they are intracellular parasites. And then for eukaryotes, we have fungi, protozoans, helminths, and viruses. So we have here this antibiotic, which is isoniazid, which only it is specific to kill mycobacteria. So then this one affects only mycobacteria. Streptomycin can kill mycobacteria, gram-negatives, and then penicillin G can kill gram-positive bacteria, and tetracycline goes from mycobacteria up to chlamydias. For eukaryotes, we have an example of an antifungal, which is ketoconazole. And then for protozoans, we have mefloquine. And then for helminths, we have niclosamide and praziquantel. Niclosamide will be for tapeworms, praziquantel for flukes. And we have this antiviral, which is specific, of course, for viruses, which is acyclovir. So arrange them in terms of broad spectrum and narrow spectrum. So which ones are narrow spectrum first?
I decided for the first one.
Isoniazid, and what else? Penicillin G. Ketoconazole, I heard that, right? Niclosamide, yes.
The acyclovir one.
Acyclovir. Praziquantel. Like quack, praziquantel. So, okay, so yes, those are narrow spectrum, and we're looking here, guys, to, of course, like groups of bacteria and viruses, okay, and fungi, of course, and protozoans. But now, what about the ones that are broad spectrum? Which ones they are?
Streptomycin, tetracycline.
Streptomycin and tetracycline will be both. And then out of the two, which one is more broad spectrum? Tetra. Now, when it comes to selection of these antibiotics, of course, acyclovir doesn't work against every single virus. So we have also some narrow spectrum within this type of antivirals or even these other antimicrobials. Like for instance, yes, tetracycline can kill mycobacteria, but it's not the drug of choice. And then tetracycline can be very good to cover gram negatives, gram positives, and chlamydias. However, if someone has a strep throat, guess what? They use penicillin G. If the person, of course, is not allergic to penicillin. And then there are some that they might have secondary effects. Okay, so we will see how we select antibiotics in just a few slides. So now let's look into how the 5 classes of antimicrobial drugs works. So just in general, we have 2 major actions for disinfectants and also for antibiotics or antimicrobials. We have those that kills microbes, and these are called cidal in the case of bacteria, bactericidal. And then we have those that prevents their growth but don't kill them. This will be static, bacteriostatic. So here we have the different modes of action and the target in the bacterial cell. So here we have a rod, and is this rod a gram positive or gram negative based on the diagram that you see there? Why? It has thick cell wall and only one plasma membrane, which will be represented by the yellow color. So then we have here the targets. And we will see details of each of these antimicrobials, but we have inhibitors of the synthesis of the cell wall. So basically, remember, no matter if it's a gram positive or if it's a gram negative, the cell wall it's a rigid structure that prevents the bacteria from being destroyed under high osmotic pressure. So then if you leave a gap here, these bacteria will burst. Okay, so that will be for inhibitors of the cell wall. Now for any bacteria, no matter which kind, they always need to produce proteins. So we have inhibitors of translation that prevents the production of proteins. So these will be called inhibitors of protein synthesis. And then, of course, the bacteria to undergo binary fission and to produce more bacteria, they have to replicate their DNA. And then we have inhibitors of nucleic acid replication and transcription, some of them, at the same time. So then you don't make a messenger RNA and you don't make 2 copies of DNA. And then we have some that produces pores in this plasma membrane, and they're basically called drugs that injures the plasma membrane as a mode of action. And then many bacteria, of course, they have enzymes, all of them actually. So they need these enzymes to produce certain items that they need or to perform certain metabolic pathways. So we have inhibitors of essential metabolites. So we will see more details about this. Classical example of inhibitors of cell wall synthesis will be penicillins. We have different kinds. Some are injected, other ones are taken orally. And specifically, we have those ones that prevent the synthesis of peptidoglycan. It will be interesting to know because now we are developing antibiotics with the aid of AI. But it will be kind of good to know if, and it can be crazy, of course, because archaea don't infect our human bodies. But it will be kind of interesting to know if there will be a way to find with AI some drugs that inhibit the synthesis of pseudomurin in the walls of these archaea and what it does to the archaea. So we'll see with time. So here we have an example of what happens to a bacteria cell when you treat it with penicillin. So here, for instance, we have gram-positive bacteria with a very huge cytoplasm enclosed, of course, by plasma membrane and cell wall before adding antibiotic, before adding penicillin. And then after you add penicillin, you see how the contents is leaking out. This is transmission electromicrograph. In the previous edition of the PowerPoint, they had a better view of this. Now they went very deep. But anyway, so we have also some drugs that affect the synthesis of proteins. And of course, the target will be the ribosomes. And then we have different types. We have chloramphenicol, erythromycin, streptomycin, and tetracyclines. Some of them, they have a subclassification based on what subunit of the 70S they affect. And at least in the case of the family members of erythromycin, which are called macrolides, these are used for when someone has allergies to penicillin. And they are very good antibiotics. They're kind of a little bit expensive, but of course, I mean, even if you don't have money, you don't want to run into the risk of using penicillin because you don't have money to use one of these macrolides in case that you have allergies. Okay, so here we have our 70S ribosomes with the 50S subunit and the 30S subunit, and then we have this little tunnel that forms in between that helps to read the messenger RNA, and then you produce your polypeptides. So if you see internally within these ribosomes, you see as the messenger RNA runs in this tunnel, and then, the ribosomes move to read this messenger RNA, you have here in this 50S portion, this tRNA, the transfer RNA, attaching every time additional amino acids, as this code dictates which amino acid come next. And then you will have some antibiotics that binds to this 50S portion and inhibits the formation of peptide bonds. So it will prevent the amino acids from linking. So examples of these antibiotics that binds to the 50S, or an example, sorry, will be chloramphenicol. And then we have some of these that prevents this tRNA from binding into the messenger RNA, forming the tRNA messenger RNA ribosome complex. These will be tetracyclines. And then we have some that changes the shape of the 30S portion, causing the messenger RNA to be read incorrectly. So in that case will be streptomycin. No matter which one it is, they will prevent the translation of messenger RNA and halting the production of proteins. In the case of some, like chloramphenicol, they have toxicity in babies and little children, so you don't give it. Otherwise, you will cause something that is called the baby Graves syndrome. So it's toxic to the bone marrow. Okay, so now for agents that damages the plasma membrane. So these are polypeptide antibiotics, and they change the permeability of the plasma membrane, so they make it more permeable. And then we have some drugs, in this case will be for antimicrobials, these polypeptide antibiotics. And then we have some that damages the membrane of fungi. So we have some antifungal drugs that combines with the membrane estrals, creating pores, like in the case of ketoconazole, for instance. And then we have some that are called ionophores that prevents the movement of cations. These are not for human use. They are used in some cattle feeds. And then here we have this antifungal destroying this yeast by creating pores in the plasma membrane. For inhibitors of nucleic acid synthesis, so we have some that can block the topoisomerase, preventing the replication of DNA. And then we have some that blocks the RNA polymerase that prevents transcription. And then we have inhibitors of these metabolites that are needed for the cells, like inhibitors of the synthesis of folic acid. Bacteria can make their own folic acid. Humans, we can't. So then, there is a pathway that allows the production of this folic acid and can be disrupted with these drugs, sulfa drugs, that competes for the binding of an enzyme that uses PABA, paraminobenzoic acid. Okay, so let's look at this animation. Very good. Let's move into. Okay, so now we're going to see why some particular drugs are not harmful to humans, and then we're going to see some advantages of using particular antibiotics, and then we're going to see why isoniazid and ethambutol are antimicrobial drugs. Okay, so that table shows you the different uses of different antimicrobials. So penicillin G and B, both of them are against gram-positive bacteria. It's highly used against strep throat infections. And then penicillin G, it is intramuscularly applied, while the other one, it requires oral administration. And then we have some penicillins that are semi-synthetic, all of these, like ampicillin, amoxicillin, that they were kind of produced because we needed something that covers more than gram-positives, like in the case of penicillins. Ampicillin, for instance, is very good, and amoxicillin against gram-negative bacteria. Okay? And then some of them has been combined with an inhibitor of penicillinase. What is penicillinase? Well, bacteria, they have their own weapon to prevent attack with these antibiotics. So sometimes they have enzymes, like in the case of penicillinase, it can destroy penicillin. And then, fortunately, we created this combination of drugs. Like, for instance, you can combine clavulanic acid with ampicillin that resists the action of this penicillinase, and then this bacteria that has penicillinase can be killed. And then we have others like carbapenems. An example of this will be imipenem, that it is used to treat infections from bacteria that are multi-drug resistant, like in the case of Pseudomonas aeruginosa. Same for monobactams, like astronam. It is also used to treat infections with Pseudomonas. And then cephalosporins, since their discovery, they have been found to be very good. And then just like phones, we have generations. So we had like cephalosporins first generation, third, fourth. We are now in the fourth generation, I think. I still believe that we are in that. Well, no, fifth, sorry. So then we have, like, for instance, this cephalosporin, which is part of the first generation that had similar spectrum as penicillin, and then this fifth generation cephalosporin that it is used to treat methicillin-resistant Staph aureus infections, or MRSA. So, examples of these will be first-generation cephalotin and then ceftaroline for the fifth. And then you expect just as fun, they get better with generations. Then the same for ceftaroline, which is fifth. And then the polypeptide antibiotics, we have this one, bacitracin, which is topical, and then vancomycin, which is a very good drug against gram-positive bacteria, which is a glycopeptide type of antibiotic. And then it is used sometimes against MRSA, and it is having some properties like penicillinase resistant. And then we have some that are called acyl deoxypeptide antibiotics like tioxibactin, that is used against bacteria gram-positive that are resistant to antibiotics. And then we have antimicrobial drugs like isoniazid and ethambutol. Both of them deal with mycolic acids, which is part of the wall of mycobacteria. But in the case of isoniazid, it prevents the synthesis of these mycolic acids, while in ethambutol, you will prevent the incorporation of these mycolic acids into the cell wall. And then, unfortunately, mycobacteria is also one of these bacteria that is highly resistant to multiple drugs, and then you use them in combination. Both isoniazid and ethambutol. And then we have these two drugs that prevent the synthesis of proteins like nitrofurantoin and chloramphenicol. Nitrofurantoin is highly used for UTIs. And then chloramphenicol is broad spectrum, but again, it causes suppression of the bone marrow in children, so it is not used in children. Aminoglycosides, we have several types. In this case, we have two oral or injected, and then we have one that it is topical, like neomycin is topical. When it comes to these other two, streptomycin and gentamicin, they have toxicity. They have toxicity for the ears and for the kidneys, so they are nephrotoxic and ototoxic. So in the case of streptomycin, it is used to treat mycobacterial infections together with ethambutol and rifampin or isoniazid. And then gentamicin, it is broad spectrum. It is used to treat infections as well with Pseudomonas. But definitely Pseudomonas, most of the times, even with that, is not susceptible. So they tend to use more these carbapenems rather than this one, the Genta. So then, since they are nephrotoxic and they are ototoxic, so they damage the kidneys and they damage the ears. Let's say that a child has an ear infection. Do you think it's a good idea to give Genta or streptomycin? More than likely it will be gentamicin, but do you think it's a good idea of a doctor prescribing gentamicin for an ear infection? No. And they don't do that. What about a person with renal failure? Their kidneys, they don't work well. Do you think it's a good idea to give an aminoglycoside? Not as well. So it's not only knowing what it kills, but also what are the secondary effects. And based on that, you choose. Okay? So you have to take into account also some secondary effects. Tetracyclines, we have different kinds. Tetracycline itself, oxytetracycline, chlortetracycline. They have certain variations in their chemical structure. All of them are good antibiotics. They are broad spectrum. And some of them, they can have some also secondary effects, toxic effects. And then they are super good to treat bacterial infections caused by chlamydias and rickettsia. So they treat intracellular parasites like these spotted fevers that we saw before. And then we have some from the glycycyclines group like tigecycline, that are broad spectrum, and they can treat MRSA and infections with Acinetobacter, which is also a multi-drug resistant microbe. And then macrolides. They have a large chemical structure, that's why they're called macrolides. We have different types: erythromycin, azithromycin, clarithromycin, and telithromycin. And again, these will be this type of antibiotics that are used as an alternative for someone who has penicillin allergy. So they are semi-synthetic, they have broad spectrum, and they have, like in the case of azithromycin, clarithromycin, they have better penetration than erythromycin. And then this telithromycin is the newest generation of these macrolides that copes with some antibacterial resistance for the other macrolides. They are, if I'm not mistaken, someone here can correct me, they are taken orally. So children are happy. Even if they're not allergic to penicillin, if they have a strep throat, of course they rather take something orally than having an IM. Okay. So for streptogramins, we have quinupristin, dalfopristin, which is known as Sinercid. So these two are the alternative to treat bursa, vancomycin-resistant Staph aureus. The transition for treatment of Staphylococcal aureus infection was initially penicillin. I mean, for decades, it was the gold standard to treat Staph aureus infections, and it was pretty good. And then they started to discover that many Staphylococcus aureus species, they didn't get killed with this penicillin, so they started to use methicillin. And then they start to see that many species or many subtypes of Staphylococcus aureus, they became resistant to methicillin, and that's how they coined that term, methicillin-resistant Staph aureus, or MRSA. And then they start to use vancomycin. And then they have vancomycin intermediate Staph aureus infections, VISA, and then they have vancomycin-resistant Staph aureus infections, which was bursa, the name of these species. So then, because of that, then they started to produce these two antibiotics, synergistics, that part of streptogramins, that they were kind of a good alternative, or they are. And then we have oxazolidinons, which one member, we have linezolid, that it is used to treat primarily infections that are resistant to too many antibiotics, gram positives. And pleuromutilins, we have mutilin and rapamycin that inhibits the growth of gram-positive bacteria. And then for agents that injures the plasma membrane, we have that daptomycin and polymyxin B. Daptomycin, it is used to treat MRSA, and then polymyxin B, it treats these gram-negative bacteria, including Pseudomonas, and these will be only topical. Inhibitors of nucleic acids. We have rifamycins, like rifampin, that inhibits the transcription of DNA, so you don't produce messenger RNA. It is used to treat tuberculosis. It has a secondary effect. It stains the body fluids orange, so you have to warn the patient. You might have an orange sweat or tears. And then in the urine, it comes really orange. And then we have these quinolones and fluoroquinolones, like nalidixic acid and ciprofloxacin, very good antibiotics, together with nitrofurantoin to treat UTIs. And then we have competitive inhibitors of the synthesis of essential metabolites. We have this combination of sulfamethoxazole and trimethoprim. So this trimethoprim, it is used to treat infections that are caused by gram-positives or gram-negatives. So here we have some examples of the different generations, differentially grouping these cephalosporins. So the first generation, which was cephalotin, was only useful against gram-positive. The second generation was more extended, covering gram-negatives like cefamandole, which is intravenously injected, and then cefaclor, which is oral. And then we have Pseudomonas aeruginosa that is treated by a third generation, and also gram-negative bacteria like ceftazidime. And then for the fourth generation, we have cefepime, which basically is useful for all the microorganisms from the previous generations, and this is intramuscular injected as well, or intravenous. And then ceftalorin, which is also useful for the previous generations, including MRSA. Any questions? Now for antifungals. So we have these drugs that affects the esterols, which is basically a lipid in the plasma membrane, which can be seen as the analog of cholesterol in our plasma membrane for animal cells. So we have polyenes like nystatin and amphotericin B. Nystatin, it is used to treat thrush, and then amphotericin B, it is used to treat systemic fungal infections, which, if you recall, these fungal infections are serious. So they go into the bloodstream, and then they spread into different tissues in the body, including the lungs, the brain, and then you have to use these strong antifungals that are only used in hospitals, actually. And they have a lot of secondary effects, especially to the kidneys. Now, for these other drugs, clotrimazole, miconazole, ketoconazole, we have some that are used topically. So we have some creams that are used to treat athlete's foot. And then we have some that can also be taken orally, like in the case of ketoconazole. And then we have these other ones, allylamines like terbinafine and naftinfinin, that are used to treat these infections that are resistant to these previous drugs. And then we have some that are called echinocandins, like capsofungin, that it is also useful to treat systemic fungal infections. And then we have inhibitors of nucleic acid synthesis. The previous one prevents the formation of the cell wall or affects the cell wall of fungi. Now, these other ones that inhibit the synthesis of nucleic acid, like fluoxetine, it is also useful for, or they are used, to treat candida infections in cryptococcus, like Cryptococcus uniformans. It also has, in this case, these ones affects the microtubules. So when the fungi is replicating, you don't have separation of these chromosomes. They have secondary effects, fluoxetine, as well. And then other ones like rhizophobin, tolnaftat, pentamidine, they also affect the microtubules. And in the case of tolnaftat, it's used to treat athlete's foot. Pentamidine, it is used to treat infections with pneumocystis carinii, which is now known as URVC. So pneumocystis pneumonia is an infection in the lungs that affects immunosuppressed patients, like patients with AIDS. Questions about antifungals? Okay, so for antiviral drugs, so we have entry and fusion inhibitors like maraviroc. This one, it is used to treat infections, like for instance HIV infection, binding to this co-receptor that is called CCR5. CCR5 receptor or co-receptor is needed for the fusion of HIV into the host cells. When it comes to HIV infections, you need to have you have to have a multiple approach so that you can prevent more cells to be infected with HIV. So then you sometimes use a cocktail of drugs, part of what is called a highly active antiretroviral therapy, or HART. And nowadays they have combined this into a single pill that has multiple drugs, so the person doesn't have to take a lot of pills or capsules and then have a lot of secondary effects, which unfortunately many of these drugs have. So we have other ones, like in the case of some that prevents the encoding of these viruses, preventing as well the integration of the genome, so they don't form a provirus, or inhibitors of the nucleic acid synthesis. So amantadine, cimetidine, these ones are used to treat influenza virus infections. They're used for treating basically the flu. And then ACT, sidovudine, tenofovir, and entricitabine, they are used to prevent the action of reverse transcriptase. So then, when you prevent reverse transcriptase, you don't form a DNA out of RNA from the HIV. Then we have these that are they're very good drugs. I mean, whenever you hear this ending clovir, like acyclovir, ganciclovir, most than likely it's a drug, or it is a drug that it is useful to treat herpes virus infections. And then we have ribavirin, lamivudine, that it is used to treat hepatitis B, hepatitis C infections. And then we have adefovir and dipovixophil that are useful to treat lamivudine-resistant infections of hepatitis B. And then we have cidofovir that is used to treat cytomegalovirus infections. So those were inhibitors of nucleic acid synthesis. In one way or the other, they prevent that from happening, and some of them prevent uncoating or the integration into the genome. Then for assembly and exit inhibitors, so they affect the maturation step and the exiting step of the viral infection. So we have nirmatrelvir, ritonavir, molnupiravir, saquinavir, zanamivir, oseltamivir, and peramivir. So the first three, they are useful to treat infections with SARS-CoV-2 infections. They are protease inhibitors as well. And in the case of ritonavir, it is a protease inhibitor for HIV. So many of these viruses, when they are forming proteins, they have basically a long polymer or messenger RNA, and they form a large polypeptide that is going to be chopped into small pieces to have specific proteins being made. So in this case, these protease inhibitors prevent this cutting of these polypeptides, so you don't have functional proteins. And then for these last three, zanamivir, oseltamivir, and peramivir, they inhibit this enzyme from influenza virus, neuraminidase. And then we have interferons, like alpha interferons, that are produced synthetically, that are useful to treat infections with hepatitis B, D, and C. So for protozoans, so we have chloroquine and mefloquine that treats malaria infections, specifically the red blood cell stage from Plasmodium vivax. And then we have artemisinin that also that it is useful for treating Plasmodium infections. We have diodohydroxyquine that it is useful to treat infections with Entamoeba histolytica, which is an amoeba. And then suramin that it is used to treat this infection with this trypanosoma that causes sleep sickness. So Trypanosoma gambiensis. Then we have metronidazole and tinidazole. These ones are used to treat Giardia lambda infections, also for Entamoeba histolytica, amoebiasis, and Trichomonas vaginalis. Metronidazole as well, guys, it causes anaerobic conditions. So it's also useful to treat infections with gram-negative bacteria. We have miltefosine that treats Naegleria fowleri infections that causes encephalitis. So it's the amoeba of the brain tissue. However, Entamoeba histolytica also can migrate from the large intestine into the liver and also can go into the brain tissue. But more specifically will be Naegleria fowleri. And then for antihelmintic drugs, we have niclosamide, which prevents the generation of ATP in the mitochondria of tapeworms. So then what it does is that the tapeworm will be all paralyzed because it doesn't have ATP. Prasequantel. So this one alters the plasma membrane of these parasites, killing flatworms. Some of these drugs, they are useful for treating infections in domestic animals, like horses, cats, dogs. Okay? So they are useful for treating infections in humans, but also in domestic animals. So praziquantel then is used to treat infections with flatworms, and then pyrantyl pamoate blocks the neuromuscular transmission in the muscles of these parasites, like roundworms, so then the parasites as well become paralyzed, they don't move. And then mebendazole, albendazole inhibits the absorption of nutrients by the parasites. It is useful to treat roundworms like Ascaris lumbricoides. This is the one that I told you that is very long, that looks like an spaghetti. And then ivermectin is useful also to treat Ascaris lumbricoides infection. So it paralyzes the intestines of these roundworms. So, the structure of penicillin. It has a nucleus that is called the beta-lactam ring. And then the different penicillins around this nucleus have different chemical structures or chemical groups that differentiates them. And then, as you saw in that animation, they prevent the cross-linking of peptidoglycans, especially in gram-positives. So then these type of antibiotics are very useful in an acute infection because the microbes are constantly replicating, forming the cell wall. So they affect more the log phase of the growth curve of bacteria. Then for natural penicillins, they are extracted from a species of penicillium. Then we saw that penicillin G is injected form versus penicillin B, which is oral. I'm hoping that we will see this graph that shows the difference in terms of how long each of these antibiotics last in the body, one versus the other one, so that you can see why one is preferred one way than the other one, and some strategies as well for treating infections with gram-positives, which is basically what natural penicillins does. So then natural penicillins, we have G and B. And then they are all susceptible to the action of penicillinases, which properly are known as beta-lactamases. And then some of these were modified, just adding some side groups, and this will be the semi-synthetic that makes them resistant to penicillinases. So we will leave it in here.